Another 3 column. Peperomia succulent plant, water storing epidermal cells. Aloe ephemeral plant, water storing mesophyll. Nerium trooxerophyte, multiple epidermis. Pinus drought avoiding coloncamatous hypodermis. Coming to the answer 1 and 3. Peperomia succulent, water storing epidermal cells is correct. Aloe ephemeral is wrong. Aloe is not ephemeral, it is a succulent plant. Nerium trooxerophyte, multiple epidermis is correct. Pinus drought avoiding is wrong. Pinus is a trooxerophyte. Coloncamatous hypodermis is correct, but it is not drought avoiding, it is a trooxerophyte. So, these are some of the important questions, or some expected questions, or some standard questions, or some stock questions regarding plant ecology. Now, this is we completed ecology with all important bits. Now, we enter the last chapter of the first year, that is genetics. Last chapter of the first year, that is genetics. So, plant genetics is the last chapter where we study Mendel importance, Mendel laws of inheritance, law of dominance, law of segregation, law of independent assortment, law of segregation, law of independent assortment, dihybrid cross, monohybrid cross, and many other aspects we discuss in this. And we also discuss the back cross, test cross, and we also discuss the probability. Probability, we discuss what is probability, what is additive law, what is products law, and many other things. Then we have linkage and crossing over with 7:1:1:7 ratio instead of 9:3:3:1 or 1:1:1:1 ratio. Now, then we discuss mutations, spontaneous mutations, and other things. All these are there in the genetics chapter. All these are there in the genetics chapter, and we discuss some important questions in this first question. An extension of probability theory used for calculating specific combination of several characters is. So, actually we use probability theory. What is probability theory? Probability means the number of a particular outcome, the number of a particular outcome in a process by the total number of outcomes in the same process. Number of outcomes in the process. In a process by the total number of outcomes. For example, capital T, small t will be there. They produce gametes. They produce capital T gamete and small t gamete during meiosis. Whenever you take the total number of gametes 2. So, what is the probability of capital T here? Probability of capital T is 1/2. Probability of small t is 1/2. So, like this probability is calculated. What is probability? Number of particular outcomes result, number of particular result in a process, number of particular outcome or particular result in a process by the total number of outcomes in a process is probability theory. This probability theory, sometimes it is extended. An extension of probability theory used for calculating specific combination of several characters in genetics is binomial theorem. It is not additive law or product law or law of independent assortment. It is binomial theorem. Next, second question. Find the wrong statement from the following. Relation between length of chromosome and number of genes present is directly proportional is wrong. Whenever the length of chromosome is more, the number of genes will be more. So, here we have to see wrong statement. Relation between length of chromosome and number of genes present is directly proportional is correct. Whenever the length is less, genes are more. Whenever the length is more, genes are less. Relation between degree of linkage and distance between the genes is inversely proportional. It is also correct. Relation between degree of linkage and distance between the genes is inversely proportional is also correct. Crossing over increases the degree of linkage. Crossing over does not increase as the degree of linkage. Crossing over always cuts the DNA or cuts the chromosome at a particular place. Exchange of bits takes place, and exchange of bits is called as crossing over. So crossing over does not increases the degree of, it decreases the degree of linkage. So third option is wrong. In a chromosome, genes are located very close to one another and mostly inherited together. In linkage, always in chromosomes genes are located very close and inherited together. So here the wrong statement is crossing over increases the degree of linkage is wrong. It must be crossing over decreases the degree of linkage. Next. This is a 3-column form. Study of following table: variety banana, Bhaskara variety of banana, spontaneous mutation, nature. Orange, novel variety of orange, induced mutations, X-rays. Lamar's evening primrose plant, induced mutations, colchicine. Rice GEB24, spontaneous mutations, nature. The correct combination is Bhaskara variety of banana is obtained by spontaneous mutations and nature is the mutant. Mutation is due to nature. Novel variety of orange is not formed by induced mutations, it is formed by spontaneous mutations only. Lamar's evening primrose is not induced mutations, it is Inothera lamarckiana, which is a type of spontaneous mutations only, not colchicine. Rice GEB24 variety, spontaneous mutations, natures is correct. So answer is 3. A and D are the answers for this question. So this is a assertion reasoning. Law of product of probabilities is applicable to dihybrid cross. Definitely law of product of probability is applicable to dihybrid cross. The phenotypic and genotypic probabilities of dihybrid cross can be calculated through product law by multiplying their individual probabilities. This is correct. Law of our dihybrid cross ratio is this one. Yellow and round are 9. Yellow and wrinkled are 3. Green and round are 3 and green and wrinkled are 1. So here the product, what is this? The phenotypic and genotypic probabilities of dihybrid cross can be calculated through product law by multiplying their individual probabilities. If you take only yellow and green color, yellow color will be 9, yellow color will be 12, 9 plus 3, green color will be 4. So 12 is to 4 is nothing but 3 is to 1. When you take only size of or nature of seed coat, round and wrinkled, again round seeds are 12 and wrinkled seeds are 4. Again 12 is to 4, it is 3 is to 1. So probability of round is, probability of yellow is 3 by 4, probability of green is 1 by 4, probability of green round is 3 by 4, probability of wrinkled is 1 by 4. So when it comes to yellow and round, 3 by 4 into 3 by 4, it becomes 9 by 16. Like this, by products law, one can easily identify the combination. So the first option is correct. A and R are correct. R is the correct explanation. So once again I will repeat this question, an important question. The law of product of probabilities is applicable to dihybrid cross is correct. Why? The phenotypic and genotypic probabilities of dihybrid cross can be calculated through product law by multiplying their individual probabilities is also correct. So both A and R are correct. R is the correct explanation of A, first option. A heterozygous round seeded plant is crossed with recessive wrinkle seeded plants. The progeny would be heterozygous round seeded plant. Heterozygous round seeded plant. Here the heterozygous round seeded plant is a parental F1 generation. It is crossed with wrinkled recessive plant and this is test cross. The test cross ratio is always 1:1. So almost 303 rounded and 303 wrinkled. It is a test cross question. Here the answer will be 1:1 and in this 1:1 is 303 rounded and 301 wrinkled, almost close to each other. So 1:1 ratio, fourth option, 303 rounded and 301 wrinkled. Now read the following and arrange the ascending order related to F2 generation dihybrid cross. Number of individuals with parental genotypes, number of individuals with dominant parental phenotypes, number of individuals with recombinant phenotypes, number of individuals with recessive phenotypes. For this the answer is less number. Here we are asking ascending order. Less number is number of individuals with recessive phenotypes, fourth option. Then third, number of individuals with recombinant phenotypes is next one. Then second, Number of individuals with dominant parental genotypes, then number of individuals with parental genotypes. So, answer is 4, 3, 2, 1. Ascending order is always 4, 3, 2, 1. This is an assertion reasoning question. A recessive phenotype can be expressed only in homozygous condition. So, recessive phenotype is always expressed in homogeneous condition like this, like this. So, recessive phenotype is always expressed in homozygous condition, small t or small y, capital R. That is correct. Dominant phenotypes can be expressed in heterozygous condition. It is impossible. Dominant phenotype cannot be expressed in heterozygous condition because it must be capital T or capital Y, capital R, small r. So, A is correct, R is false. So, a recessive phenotype can be expressed only in homozygous condition is correct. Dominant phenotypes can be expressed in heterozygous condition is wrong. So, A is true, R is false. Now, read the following table and identify the correct match related to test cross progeny of Buttison-Punnett experiment with seat P. Buttison's test cross experiment is nothing but linkage. So, read the following table with identify the correct match related to test cross progeny of Buttison and Punnett's experiment is nothing but linkage. Buttison and Punnett conducted linkage in suite B. They have obtained one F1 progeny and then linked it with or crossed it with homozygous or recessive pair. And blue flowers and long pollen, blue flowers and round pollen, red flowers and long pollen, red flowers and round pollen. Answer is 4. Blue flowers and long pollen 192. Blue flower and round pollen, blue flower and round pollen 23. Blue flower and long pollen 182, 30 and red flower and round pollen is 182. So, answer is 4. A4. Blue flower and long pollen grain is 192. Blue flower and round pollen is 23. Red flower and long pollen is only 23. Red flower and round pollen is 192. So, 182. So, the ratio will be 7:1:1:7. So, for 8th question the answer is 4. Read the following statements and identify the correct statement. Backcross involves crossing between F1 hybrid and either of the parents. It is correct. Backcross is small t which is crossed with capital T or small t. This is correct. This is backcross. Test cross involves crossing between F1 hybrid and its recessive parent. This is also correct. This is also correct. All test crosses are back crosses. All test crosses are back crosses. It is also correct. All test crosses are back crosses. All back crosses are test crosses. So, answer is 1, 2, 3, 4. All are correct statements here. Next. Assertion reasoning. Linkage can help in maintaining genetic integrity of organisms to a large extent of several generations. Genes, when present very closely on chromosomes, they get inherited together. When genes controlling all dominant characters are present very close to each other, they inherited together generation after generation, generation after generation. So, any advantageous character present in an organism can be retained for many generations. This is assertion. Linkage can help in maintaining genetic integrity of organisms to a large extent for several generations. Whenever dominant genes or recessive genes controlling same character are present close to each other, they are easily inherited generations after generation. Reason: linkage reduces the possibility of variability in gametes unless crossing over takes place. So, linkage reduces the possibility of variability. Because the genes are not getting independently assorted, that new characters will come in lesser amounts. We do not say new characters will not come, new characters come in lesser amounts, and unless crossing over takes place, linkage cannot be minimized. So, assertion and reasoning both are correct. Reason is the correct explanation of assertion. So, answer is both A and R are correct. R is the correct explanation of A. In a backcross, P1 generation plants are crossed with P1 generation plants. Wrong. It is not capital T, capital T into small t, small t. This is wrong. This is not a back cross. F1 generation plants are crossed with any one of the P1 generation, parental 1 generation. F1 generation means capital T, small t. It may be crossed with capital T or small t, small t. This is back cross. Then third option. F2 generation plants are crossed with F1 generation individuals. It is 100% wrong. F2 generation plant cannot be crossed with F1 generation plant. Cross between two different F1 individuals is wrong. So, in a back cross, the correct is F1 generation plants are crossed with any one of the parental generation plants, that is either dominant parent or recessive parent. So, second option. In terms of probability, half obtained in the selfing of F1 monohybrid indicates 1 by 2. 1 by 2 is capital T, small t when combines with capital T, 4 options appear. Capital T, capital T, capital T, small t, capital T, small t, small t, small t. The probability here, probability of capital T is 1 by 4. The probability of capital T, small t is 2 by 4 or 1 by 2. The probability of small t is 1 by 4. So, here the question is, in terms of probability, 1 by 2 half obtained in the selfing of F1 hybrid indicates probability of a gamete with T allele. This is correct. Probability of gamete with small t allele, correct. Probability of an individual with capital T genotype, probability of individual with small t genotype is wrong. So, answer is 1 and 2 only. In terms of probability, half obtained from the selfing of F1 monohybrid indicates probability of gamete with a capital T allele, probability of gamete with a small t allele. So, first option. Now, assertion reasoning. This is a big question, follow it carefully. In a Mendel dihybrid cross, F2 progeny showed not only the parental combinations yellow and round and green and wrinkled, but also shown the new combinations like yellow wrinkled and green round phenotypes. It is correct. In Mendel's dihybrid cross, 9:3:3:1 ratio is obtained. 9 are yellow round, 3 are yellow wrinkled, 3 are green round, 1 is green wrinkled. So, F2 progeny showed not only parental combinations yellow and round and green and wrinkled, but also the new combinations like yellow wrinkled and green round phenotypes. What is the reason for this? These new combinations are produced due to genetic recombination among the factors by independent assortment. Factors means genes. So, these new combinations are obtained because the genes get separated independently, independent assortment occurred, they again fused a like form of gametes and then produced a new combination. So, that is law of independent assortment. So, both A and R are true. R is the correct explanation of A. First option is for this question. What will be the phenotypic ratio of offspring obtained by dihybrid cross? Dihybrid test cross, phenotypes obtained, phenotypic ratio of offspring obtained by dihybrid test cross. Dihybrid test cross means capital Y, capital R, small r into small y, small r, small r. If you draw a checkerboard or Punnett square, it comes like this. Capital Y, capital R, capital Y, small r, small y, capital R, small y, small r. Here the genes will be only capital Y, small r. So, YY, RR, yellow round, dominant is there. Capital Y, small r, yellow wrinkled. Small y, capital R, small r, green round. Small y, small r, green wrinkled. So, in any dihybrid test cross, the ratio is 1:1:1:1. So, dihybrid test cross always results in a ratio called 1:1:1:1. So, answer is 2, 1:1:1:1. Now, this is a matching. Contrasting characters, genotype, alleles, gene locus, Punnett square. Gene locus, Punnett square. The alternative forms of a character, the genetic makeup of an individual, the two alternative forms of a gene, the location of a particular gene on a given chromosome. All possible combinations or unions of different male and female gametes during fertilization are represented by this. Answer is second. A is 1. 1 is contrasting characters. The alternative forms of a character is contrasting character. B is 3. What is genotype? The two alternative forms of a gene is genotype. C 2. So B is 2. Genotype. The genetic makeup of an individual is called genotype. Alleles. The two alternative forms of genes are called alleles. Gene locus. The location of particular gene on a given chromosome is correct. Punnett square. All possible combinations or unions of the different male and female gametes during fertilization are represented by this. So contrasting characters, first option. Genotype, second option. Alleles, third option. D, gene locus, fourth option, and Punnett square, fifth option. So all are given in a correct order. So A1, B2, C3, D4, E5. So contrasting characters, alternative forms of a character. Genotype, the genetic makeup of an individual. Alleles, the two alternative forms of a gene. Gene locus, the location of a particular gene on a given chromosome, and Punnett square, all possible combinations or unions are given in the form of special blocks, which was discovered by Reginald C. Punnett. So answer is second option. Now a matching. List 1, list 2. Agents of mutations, ethyl methane sulfonate, largely used in crop improvement and physical mutagens. First option. A3. Agents of mutations, mutagens program. This is called mutagen program. EMS, B1. Ethyl methane sulfonate. C. Largely used in crop improvement. Chemical mutagens are widely used in crop improvements. And D4. Physical mutagen, widely used in the improvement of crop varieties. So D4. Answer is 1. Another matching. Dimutation theory, GEB24, neutrons, nitrous acid. Spontaneous mutant variety of rice, chemical mutagen, Hugo de Vries, physical mutagen, somatic mutant. Answer is 3. A3. Dimutation theory was written by Hugo de Vries. B1. GEB24 is a spontaneous mutant variety of rice. C4. Neurons, physical mutagens. Neutrons are physical mutagens. And D2. Nitrous acid is a chemical mutagen. Here there is no somatic mutant, all the 4 are given. So dimutation theory is Hugo de Vries. GEB24 is spontaneous mutant variety of rice. Neutrons are physical mutagens and nitrous acid is a chemical mutagen. This is answer is 3. Which one of the following set of answers represent the correct ascending order related to F2 progeny in a dihybrid cross of round yellow and wrinkled green obtained by Mendel? Green wrinkled, only 1. Green round with YYRR is less. Yellow wrinkled is less. Yellow round is more. So answer is 3, 1, 4, 2. I will repeat this question. Which one of the following set of answers represent the correct ascending order related to F2 progeny in dihybrid cross? 9:3:3:1 progeny in dihybrid cross of round yellow, that is yellow round and green wrinkled obtained by Mendel. Less is green wrinkled. Less is green wrinkled, 3, because only 1. Next is green round with capital Y, small y, capital R, small r will be less. Only 2 will be there. Then 4. Yellow wrinkled are 3 around. So 1, 2, 3, 1, 2, 3, 9 is the answer. So answer is 3, 1, 4, 2. Third option. Now a 3-column match. Study the following table and incorrect combinations. It is not correct combinations, it is incorrect combinations. De Vries mutations, Inothera lamarckiana. De Vries mutations, Inothera lamarckiana is correct. Morgan linkage Giamese is correct. Morgan linkage Giamese is correct because Morgan discovered linkage in both Drosophila and Giamese. Bedson and Punnett crossing over Drosophila is wrong. Bedson and Punnett conducted linkage experiments in Drosophila, not crossing over experiments. Reginald C. Punnett chromosomal maps Pisum sativum is wrong. Reginald C. Punnett is not chromosomal map. Reginald C. Punnett is known for Punnett square or what you called checker board. So incorrect combinations are 3 and 4. Hugo de Vries mutations Inothera lamarckiana is correct. Morgan linkage Giamese is correct. Bateson and Punnett is not crossing over, it is linkage. Reginald C. Punnett is chromosomal maps is correct. Chromosomal maps itself is wrong because he discovered Punnett square or checkerboard. So 3 and 4 are incorrect combinations. When F1 hybrids are test crossed, if the phenotypic ratio is 7:1:1:7, it indicates. When F1 hybrid is test crossed, if the phenotypic ratio is 7:1:1:7 obtained, this indicates linkage only. And what is the answer means? Dominant alleles and recessive alleles segregate during gamete formation is wrong. Dominant alleles and recessive alleles cross over each other in most of the cases is wrong. Dominant alleles and recessive alleles tend to pass together in the same gamete is correct, that is linkage. So dominant alleles and recessive alleles tend to pass together in the same gamete is correct. Dominant alleles and recessive alleles combine independently into different groups is wrong. So 7:1:1:7 is a linkage concept. Dominant alleles and recessive alleles segregate during gamete formation is wrong. Dominant alleles and recessive alleles cross over each other in most of the cases is wrong. Dominant alleles and recessive alleles tend to pass together in the same gamete is correct, that is linkage. And dominant alleles, recessive alleles combine independently is in different groups is wrong. So here the correct option is third option. Now, a typical Mendelian dihybrid cross, the total number of F2 plants is 1600. A typical Mendelian dihybrid cross, the total number of plants is 1600. From this, calculate the number of double heterozygous plants, double recessive plants, homozygous double dominants and double dominants respectively. Double heterozygous plants will be only 400. Double heterozygous plants will be 400. Double recessive means small y, small r, only 1, so 100. Homozygous double dominants, for this you have to remember the phenotypic ratio 1:, genotypic ratio 1:2:4:1:2, 1:2:1:1. This is genotypic ratio of dihybrid cross. You have to remember this and the combination of genes, then you can write this type of questions. So in a typical Mendelian dihybrid cross, the total number of F2 plants obtained are 1600. That means 9:3:3:1, 16. 1600 plants. So 9:3:3:1 we have to calculate. So from this, calculate the number of double heterozygous plant. Double heterozygous plant means capital Y, capital R, small r. Only 4 will come. Capital Y, capital R, small r. Only 4 will come. So they are 400. Double recessive plants, only 1 will come, 100. Homozygous double dominants, only 1 will come, 100. And double dominant capital Y, capital R, 9 will come, that is 900. So ratio or answer is 400, 100, 100, 900. First option. Experimental material for Bateson and Punnett. Experimental material, Bateson and Punnett is known for linkage experiments in Drosophila and wild pea plants. Wild pea plant, Pisum sativum is a garden pea. Lathyrus odoratus is wild pea, Lathyrus sativum is wrong, Drosophila is wrong. So experimental material for Bateson and Punnett is Lathyrus odoratus. So second option. Which of the following is a chemical mutagen? X-rays, alpha rays, gamma rays, ultraviolet rays, neutrons, all comes under physical mutagens. Chemical mutagens that help in mutations are colchicine, mustard gas, nitrous acid, ethyl methane sulfonate, EMS and MMS, ethyl methane sulfonate, formaldehyde, formaldehyde, ethyl methane sulfonate, formaldehyde. So these are some of the chemical mutagens which induce mutations by adding them: colchicine, mustard gas, nitrous acid, ethyl methyl sulfonate, methyl methane sulfonate, formaldehyde, all are chemical mutagens. So here the chemical mutagen given is EMS, ethyl methane sulfonate. Next, the exchange of non-sister chromatids between homologous chromosomes is called. The exchange of non-sister chromatids between homologous chromosomes is called crossing over. It is not linkage. What is linkage? Many genes coming together along with gametes is linkage. Dihybrid cross, monohybrid cross is wrong. So the exchange of non-sister chromatids between homologous chromosomes is called crossing over, which we discussed in meiosis chapter during patch 18. So answer is crossing over. The plant which became famous due to theory of mutations. The plant which became very famous due to theory of mutations is not Triticum vulgare, wheat, not Pisum sativum. Pisum sativum is made popular by Mendel's laws of inheritance, monohybrid cross, dihybrid cross. Primula sinensis is also wrong. Inothera lamarckiana, evening primrose plant, is very popular plant which became very famous due to theory of mutations. In this plant only DeVries conducted or DeVries discovered mutations. The coexistence of two or more genes, two as is wrong, two or more genes in the same chromosome is called not crossing over, not dihybrid cross, not monohybrid cross, but linkage. So the coexistence of two or more genes in the same chromosome is called linkage. Next, identify the mismatch in the following: mutations, spontaneous mutations. Spontaneous mutations occur naturally. The occurrence is very slow and rare. 1 in 1 lakh may show this mutation. But there are some achievements. Bhaskara variety of banana, delicious apple, novel variety of orange, and GEB24 variety of rice. GEB24 variety of rice. So spontaneous mutations means the mutations which occur very slowly and naturally. The occurrence is very rare and very slow. 1 in 1 lakh may show these mutations. The best examples are Bhaskara variety of banana, delicious variety of apple, novel variety of orange, and GEB24 variety of rice. Here Bhaskara variety of banana is correct. Novel variety of orange is correct. But delicious variety of mango is wrong. Delicious variety of apple is correct. So GEB24 variety of rice is correct. So what is the mismatch in the following is delicious mango. Delicious mango is wrong. It is delicious apple. Study the following regarding dihybrid F2 generation and arrange these as per ascending order the number of individuals. Number of individuals with parental genotypes, number of individuals with F1 phenotypes, number of individuals with double homozygous genotypes, number of individuals with single homozygous phenotypes. Answer is first ascending order A. Number of individuals with parental genotypes will be less. Next comes number of individuals with double homozygous genotypes. Then comes number of individuals with single homozygous genotypes, and finally it is number of individuals with F1 phenotype. F1 phenotype means capital Y, capital R, small r. So number of individual with parental genotypes are only 2. Capital Y, capital R, small y, small r are only 2. So first always A. Number of individuals with F1 phenotype comes in the last. Number of individuals with double homozygous genotypes is after number of individuals with parental genotypes, and number of individuals with single homozygous genotypes is, so answer is A, C, D, B. A comes first, C comes next, D comes then, B comes next. So study the following regarding dihybrid F2 generation and arrange these as per ascending order of number of individuals means A, C, D, B. In a dihybrid cross, if the four phenotypic combinations of F1 offsprings are formed in equal proportions, the genotype of the parents will be. So, in a dihybrid cross, if the four phenotypic combination of F1 offsprings are equal proportion means capital Y, small y, capital R, small r, when crossed with small y, small r, the ratio will be 1:1:1:1. This is called dihybrid test cross. So, the question is, in a dihybrid cross, if the four phenotypic combinations of F1 offsprings are formed in equal proportions, the genotype of parent should be capital R, capital Y, small y into small r, capital Y. So, this is nothing but dihybrid test cross. Then only phenotypic ratio is 1:1:1:1. So, for 25th question, the total explanation is to tell whether what type of cross it is. We have to identify as in a dihybrid cross, if the four phenotypic combinations of F1 offsprings are formed in equal proportions, that means 1:1:1:1 ratio, the genotype of parents should be capital R, capital Y, small y into small r, small y, which is nothing but test cross. Arrange the following chromosomal number in ascending sequence: trisomic, nullisomic, tetrasomic, monosomic. What is monosomic? 2n-1. What is trisomic? 2n+1. What is tetrasomic? 2n+2. What is nullisomic? 2n-2. So, this is a combination of change in the chromosomal number. Monosomic 2n-1, trisomic 2n+1, tetrasomic 2n+2, nullisomic 2n-2. This comes in the mutations change in the chromosomal number. For this, the answer is nullisomic 2n-2, then monosomic 2n-1, then trisomic 2n+1, and tetrasomic 2n+2. So, arrange the chromosomal number in ascending sequence in trisomic, nullisomic, tetrasomic, monosomic individual is nullisomic, monosomic. Nullisomic is 2n-2, monosomic is 2n-1, trisomic is 2n+1, tetrasomic is 2n+2. So, answer is second one: nullisomic, monosomic, tetrasomic and trisomic and tetrasomic. In a cross between AABB and AABB, capital A, capital B, capital A, capital A, capital B, capital B, and small a, small b, small a, small a, and small b, the ratio of AABB, all capitals, the ratio of capital A, capital A, capital B, small b, the ratio of small a, small b, capital B, small b, and ratio of AABB, all small in F2 generation will be definitely 1:2:2:1. This is the ratio 1:2:2:1. This is a assertion reasoning question. Closer genes are linked strongly while farther genes are linked weakly. It is correct. Closer genes are linked strongly while farther genes ante genes away from them are linked weakly. The degree of linkage between genes depends upon the distance between the loci. Loci means the place where genes are attached or arranged. So, because the degree of linkage between the genes depends upon the distance between the loci only, closer genes are linked strongly while the farther genes are linked weakly. So, A and R are correct. R is the correct explanation of A, first option. When a chromosome with genes in order of MNOPQRST may change to MNOP, MNO, PQRST, this change is due to the phenomenon. There are 4 combinations. Deletion, a part of the chromosome is lost. Duplication, a part of the chromosome is doubled. Inversion, the chromosomal genes are attached inversely. And finally, translocation, a part of the chromosome is lost but go and attaches to some other non-homologous chromosome. So, deletion, a part is lost. Some parts get doubles, arranged in reverse direction, and it goes and attaches to non-homologous part of a chromosome. Here in this, there are some genes which are duplicated. MNOP, again MNOPQRST. So, MNO is duplicated. So, definitely it is not translocation, it is not deletion, it is not inversion, it is duplication. See the question. When a chromosome with genes in an order MNOPQRST is the normal order, may change to MNOP and again MNOPQRST. So, MNOP is repeated. So, when genes are repeated, it is called duplication. So, fourth option. Spontaneous mutations occur without any mutagenic agent. Spontaneous mutations occurs without any mutagenic agent. Individual mutations are caused mutagenic agents. Second option is correct. Spontaneous mutations occur without any mutagenic agent is wrong. Nature must be some mutation, something must be given by nature. So, spontaneous mutations occur without any mutagenic agent is wrong. Individual mutations are caused by mutagenic agent is correct. So, A is false, R is true. F2 generation of monohybrid cross of a tall plant resulted, tall and dwarf plant resulted in 200 plants. Number of tall and dwarf plants. If you cross capital T with small t, the resultant F1 generation is capital T, small t. This is dominant tall and this is dominant recessive. If the same thing is taken for F2 generation, the phenotypic ratio will be 3:1. So, 3 will be tall and 1 will be dwarf. Here 200 plants are taken. So, definitely 150 will be tall and 50 will be dwarf. So, when a capital T dominant tall is crossed with small t recessive dwarf, F1 generation is all capital T, small t, tall. When it comes to F2 generation, the ratio is capital T, small t, capital T, small t, small t, small t. Phenotypic ratio is 3:1, genotypic ratio is 1:2:1. The question is, F2 generation of monohybrid cross of a tall and dwarf plant resulted in 200 plants. Number of tall and dwarf plants are, if the ratio is 3:1, the answer will be 150 and 50. Identify the true statement. Chromosomal maps are useful to identify the distance between genes in a linkage group. Chromosomal maps expresses the percentage of recombinations. Chromosomal maps are useful to estimate the phenotypic ratio. Chromosomal maps designed by Morgan, Bateson and Punnett. Chromosomal maps are useful to identify distance of genes in a linkage group is correct. Chromosomal maps expressed in the percentage of recombinations is correct. But chromosomal maps are not useful in estimate the phenotypic ratio. So, answer is 1 and 2. 3 will not come in the chromosomal maps because it is not useful to estimate the phenotypic ratio. So, 1 and 2 is correct. So, match the following and select the correct option. 1:1, 3:1, 1:1:1, 1:2:1, 1:3:3:1, monohybrid genotypic ratio in F2, monohybrid cross in F2, incomplete dominance, dihybrid test cross in F2, dihybrid test cross, monohybrid test cross. Answer is 2. A is 6, monohybrid test cross 1:1. B is 2, 3:1 is monohybrid cross in F2. Then C is 5. What is C? 1:1:1:1. 5 is dihybrid test cross. D is 3, 1:2:1. What is 3? Incomplete dominance. And E is 4. What is E? 9:3:3:1 dihybrid cross in F2. So 1:1, 3:1, 1:1:1:1, 1:2:1, 9:3:3:1 are the ratios, and for that link A2, A4, B2, C5, D1, E4 will be the answer for this. So we have to identify all the ratios and for what that ratio is represented for. So second option. Statements, read the statement and find the correct one. Crossing F1 hybrid with recessive parent is called test cross is correct. Crossing of a dihybrid to a double recessive parental plant with 2 factors is test cross is correct. Test cross is useful to recognize the genotype of an individual with dominant characters is correct. Test cross is done whether the resultant one is a heterozygous parent or homozygous parent. So all the 3 are correct. Crossing of F2 hybrid with the recessive parents is called test cross is correct. Crossing of dihybrid to double recessive parent is called 2-factor test cross is correct. Test cross is useful to recognize the genotype of an individual with dominant characters is correct. So what is the correct combination? Means all are correct. Second option. Now this is a matching. Match the following and choose the correct options. Bhaskara banana, just now I told spontaneous mutation. So A is 2. A is 3. Sorry. X-rays B2, physical mutagens, X-rays, gamma rays, alpha rays, neutrons, all comes under physical mutagen. C, colchicine, C is 1, chemical mutagen. Mutations occurs in DNA fragments, D is 5, point mutations. When a mutation occurs in a small bit of DNA or near the 2 nucleotides, it is called point mutations. So Bhaskara banana is a spontaneous mutation variety. X-rays are physical mutagens, colchicine is a chemical mutagen. Mutations occurring in DNA fragments is popularly called point mutations. Second option. Now coming to the last question. Progeny with 7:1:1:1:7 was obtained instead of 1:1:1:1 with a dihybrid test cross. The reasons for more number of parental combinations and less number of recombinations respectively in this progeny are. Why? Because it is linkage and crossing over, not homozygosity or recessive characters, dominant and recessive characters, chromosomal mutations or gene mutations. So progeny with 7:1:1:7 was obtained instead of 1:1:1:1. Normally dihybrid test cross means result must be 1:1:1:1. But here a special ratio has obtained, that is 7:1:1:7. It is due to linkage and crossing over.